Grandpa's blue eyes and my dimples. And you get your height from your father and your mother's fair complexion.
I wonder why I get these features from different family members.
By the end of this lesson, you will be able to define heredity, define variation, describe the life and work of Mendel, describe the terms used in genetics, describe Mendel's experimental study with pea plants, describe the process of sex determination. This boy resembles his mother in skin color and father in height. Let's see why. All the cells in his father and mother have 46 chromosomes each. During the reproductive process, his father produces a male gamete and his mother produces a female gamete, each with 23 chromosomes. The male gamete fuses with the female gamete to give birth to a child. So, the newborn child receives 23 chromosomes from the father and the other 23 from the mother. Boy receives genes for the characters from both the parents. The boy has received the gene for height from his father and the gene for skin color from his mother. That's why the boy resembles his parents in skin, color and height. But the boy differs from parents in other ways, such as his curly hair and blue eyes, which neither of the parents have. Any characteristic that is transferred from parent to offspring, such as height and color, is known as a trait. As this trait is inherited from the parents, it is known as an inherited trait. This process of passing traits from parent to offspring is called heredity. Offspring are never an exact copy of their parents, as you have seen in this case. There may be some differences. Curly hair and blue eyes of the boy are the differences that are called variations. Gregor Johann Mendel was the pioneer among geneticists who discovered the concept of inheritance of characters or traits from parents to offspring. He proposed the principle of inheritance and is known as the father of genetics. He experimented with pea plants and found variations among them. Later, these variations were found to be genetic variations. Variations in heredity occur during sexual as well as asexual reproduction. Let's look at a few examples. When we observe a field of sugarcane, we hardly find variations among the individual plants. A bacterium divides into 2 bacteria, and the resultant 2 bacteria redivide to generate 4 bacteria. All these bacteria are identical. However, there may be minor differences generated due to small inaccuracies in DNA copying. Now things change slightly in the case of sexual reproduction. Maximum variation is produced by this process, as can be seen in the case of human beings. Did you know that Mendel was educated in a monastery and studied science and mathematics at the University of Vienna. He wanted to be a teacher, but he failed the teaching examination. However, he did not lose hope. He returned to the monastery and started growing pea plants in the garden. Mendel blended his knowledge of science and mathematics to keep count of the pea plants exhibiting a particular feature in each generation. Mendel researched heredity and arrived at the laws of inheritance. He used a number of pea plants with various traits such as round or wrinkled seeds, tall or short plants, white or violet flowers, and so on. Let's look at some of the most important terms in heredity and variation. The word gene comes first. It is the DNA segment of the chromosome. Genes control the expression of characters. Genes were called factors by Mendel. Each gene is present in two alternative forms called allele. Each allele controls single traits. Tallness in a plant is a dominant trait, controlled by a dominant allele, and is represented by the uppercase T. Shortness in a plant is a recessive trait, controlled by a recessive allele, and is represented by the lowercase t. These alleles combine to make a genotype such as uppercase TT or uppercase T and lowercase t or lowercase tt. Uppercase TT and lowercase tt are homozygous, and uppercase T and lowercase t is referred to as heterozygous. Yet another important term is a phenotype. Phenotype is the physical appearance resulting from a genotype. For example, if a particular plant is tall, it is the tall phenotype. Punnett square helps to predict the possible genotypes and phenotypes of offsprings. Let's see how to make a Punnett square. Place the genetic combinations of the parents, like parent lowercase tt, along the right of a grid, and other parent uppercase tt along the left of the grid, and the alleles of the gamete uppercase T and lowercase t below the parents. Combine the parent alleles and place the offspring's possible genetic combinations in the below box. F1 generation is the first filial generation offspring produced by crossing two parental strains. F2 generation is the second filial generation offspring produced by crossing F1s.
I don't think I've understood completely.
Let's take another example to make things clear. The human population has two variants of earlobes. Some have free earlobes, while others have attached earlobes. A particular mechanism is responsible for such variations. The inheritance of traits is related to the fact that both the father and the mother contribute equal amounts of genetic material to the child. If we try to correlate the earlobe of a child with that of his parents, we understand that the father's gene for earlobe is dominant, because of which the child developed an attached earlobe. Well, let me explain this through Mendel's experiment, in which inheritance of only one character, for instance the height of the pea plant, is considered. Such inheritance pattern is referred to as monohybrid inheritance. The Punnett square shows a cross between parental tall uppercase TT and dwarf lowercase tt plants. The gametes produced by them are T and T, and the F1 generation has all uppercase T and lowercase t progeny. In first generation or F1 progeny, all of the plants were tall. This means that only one of the parental traits were visible. The trait which is visible in F1 generation is called the dominant trait, and one which remains suppressed is known as the recessive trait. Here, tallness is the dominant trait and shortness is the recessive trait.
Were all the tall plants in the F1 generation exactly as tall as the plants of the parent generation?
Yes, they were all as tall as the parent generation. Next, Mendel allowed the F1 tall plant of the genotype uppercase T and lowercase t to reproduce by self-pollination. As a result of random fertilization, the resultant zygotes can be of the genotypes uppercase TT or uppercase T and lowercase t or lowercase tt. Interestingly enough, the second generation or F2 progeny were not all tall. Instead, 1/4 of them were short. This indicates that both the tallness and shortness traits were inherited in the F2 plants. Now, let's see the entire experiment in the form of genotypic and phenotypic representation. From the Punnett square, it is seen that random fertilizations result in 1/4 uppercase TT, half uppercase T and lowercase t, and 1/4 lowercase tt. This gave the genotypic ratio of 1:2:1. 1 pure tall plant, 2 hybrid tall plants, and 1 dwarf plant. Thus, 75% of the progeny produced in F2 generation were tall and 25% were dwarf. This gave the phenotypic ratio of 3:1.
What happens when pea plants of two different characteristics are bred with each other?
That's an interesting question. Let's consider a hypothetical situation. We cross a plant producing round and yellow seeds, uppercase RR and uppercase YY, with a plant producing wrinkled green seeds, lowercase RR and lowercase YY. The inheritance of two such characters is known as dihybrid inheritance. The entire F1 progeny produces round and yellow seeds, uppercase R and lowercase r, and uppercase Y and lowercase y. In such a case, round and yellow seeds would be the dominant traits.
What happens when these plants of the F1 progeny are self-pollinated?
Mendel found that some plants of the F2 progeny were similar to their parents and produced round yellow seeds, and some of them produced wrinkled green seeds. However, some plants of the F2 progeny even showed new combinations like round green and wrinkled yellow seeds. Therefore, it can be safely concluded that the round or wrinkled trait and the yellow or green colored traits are independently inherited.
How do the traits get expressed? If cellular DNA is the information source for making proteins in the cell, how do proteins control the characteristics?
Well, consider a plant with tallness as a characteristic. We know that plants have hormones that trigger the growth. Assume that an enzyme performs this process. If this enzyme works efficiently, a large quantity of hormone will be made, and the plant will be tall. However, if the gene for that enzyme has an alteration that makes the enzyme less efficient, the amount of hormone will be less, and the plant will be short. Thus, genes control characteristics or traits.
Why do we have only boys in our family? Why not girls? Does such a difference occur only in humans?
The sex of human beings is determined by the sex chromosome. In some animals like crocodiles, the temperature at which fertilized eggs are kept determines whether the animals developing in the eggs will be male or female. In other species, such as snails, individuals can change the sex depending upon the external environment. It indicates that the sex is not determined by germ cells. Let's see how genetic inheritance determines the sex. Genes are present not as a single long thread of DNA, but as separate independent pieces called chromosomes. All humans have 23 pairs of chromosomes. Of these, 22 pairs of chromosomes have a maternal and a paternal copy. But one pair, called the sex chromosome, is odd. Women have a perfect pair of sex chromosomes called uppercase XX. But men have a mismatched pair in which one is a normal-sized X, while the other is a short one called Y. So, women have XX, while men have XY chromosomes. Let's look at the inheritance pattern of the uppercase X and uppercase Y chromosomes. All children will inherit an X chromosome from their mother, regardless of whether they are boys or girls. A child who inherits an X chromosome from her father will be a girl, and one who inherits a Y chromosome from him will be a boy.
There's my brother, and that is my cousin next to me. These are my parents, and those are my grandparents.
Sarah and her brother both have freckles on their faces and wear glasses. As you can see, They get their freckles and their eyesight from their parents. This is because Sarah and her brother have common ancestors, their grandparents. This is an example of the fact that all life on Earth is descended from a common ancestor. So we can say that biological evolution is descent with modification. This lesson explains how species are formed and how they evolve over generations. At the end of this lesson, you will be able to define evolution, explain the mechanism of evolution, define variation, explain the mechanism of evolutionary changes, define species, define speciation, and describe the process of speciation. Evolution is defined as a genetic change in a population that is inherited over several generations. The question is, in what manner are these genetic differences passed on? Some researchers believed life developed in a linear fashion, from simple forms to complex forms. Charles Robert Darwin had a different theory of evolution. He set out on a voyage, observed various species of life on Earth, and put forward the idea of evolution of species by natural selection. Darwin compared the emergence of new species to the branching of a tree. He said that a species inherits its characters from its ancestors. To understand evolution, let's look at some examples. These are two different beetle populations. Both these species of beetles usually feed on the leaves of a particular bush. We will see which of these beetle populations is an example of evolution. The bush gets infested with a disease, and the amount of leaf material available to the beetles is reduced. So, during the course of generations, the average weight of the beetles decreases. When the plant disease is eliminated, the average weight of the beetles returns to normal. Since the change is not inherited over generations, this is not an example of evolution. Now, look at this sample population of two types of beetles. Around 90% of the beetles are green and 10% are brown. This means 90% have genes that provide the color green and 10% have genes that provide the color brown. Generations later, this is what the sample population looks like. Brown beetles make up about 70% of the population. These two generations of the same population are genetically different, showing an evolutionary change. Changes in genes of color variation occur and are inherited over generations. So, this is an example of evolution. Evolution requires genetic variation. Variation is the differences among individuals of a species caused by genetic and environmental factors. Let us discuss this with the same beetle example. A green beetle population turning into brown beetles through an evolutionary change is called a mutation. A mutation is a change in the sequence of nitrogen base pairs in the DNA. A mutation in the gene for green coloration in green beetles leads to offspring with brown coloration. There may be a situation where some brown beetles join a population of green beetles. This is called migration. Breeding between the brown and green beetles introduces new gene combinations into the population. This movement of genes from brown beetles to green beetles is called gene flow. Let's consider a slightly different situation. A population of green and brown beetles is crawling on the leaves of a bush when a bird appears. Brown beetles are more visible on the bushes than green beetles, so the bird is more likely to feed on them. Green beetles, which are likely to survive, will bear the responsibility of producing the offspring. They will pass their genes for green coloration onto their offspring. Therefore, the green beetle is better adapted to the environment than the brown beetle. This process, in which organisms are best suited to their environment, is called natural selection. Now, let's talk about another situation. In a sample of green and brown beetles, several green beetles are killed when a man steps onto the bushes where the beetles are feeding. This results in a greater number of brown beetles in the next generation. So, accidents in small populations can change the frequency of some genes in a population. This genetic variation in small populations caused by an environmental factor is called a genetic drift. Let's see how the speciation happens. Before that, let's understand what a species is. Take a look at the spiders. Each spider looks different, but they are all able to interbreed. A species is defined as a group of individuals that can interbreed in nature. Thus, we can say that these spiders belong to the same species. Speciation is an event that splits a population into two independent species which cannot reproduce with each other. Let's see a simplified scenario of speciation by geographic isolation. Here is a population of beetles laying their eggs on bananas. If a hurricane strikes the mainland, and the bananas with the beetle eggs on them are washed away to an island, it is called genetic drift. The beetles hatch out of the eggs and mature on the island. Ecological conditions are slightly different on the island as compared to the mainland. This leads to a change in morphology and food preferences in the beetles over the course of generations. In case another storm occurs, the beetles from the island may get washed back to the mainland. However, the beetles will not breed with the mainland beetles any longer, since they've evolved different behavioral characteristics by now. The few that do mate with the mainland beetles produce inviable eggs because of the genetic differences between the two populations. The lineage has split now. Genes cannot flow between the populations anymore. This process is referred to as speciation. Millions of years ago, on Earth, lived huge gigantic animals called dinosaurs. In the process of evolution, all these huge gigantic animals became extinct. Using the remnants of these dead animals to compare against the characteristics of existing animals, evolutionary relationships were developed. This is how we came to know about the existence of dinosaurs. However, there are several other pieces of evidences of evolution that suggest a link between the different species. In this lesson, you will learn about evolutionary relationships and human evolution. At the end of this lesson, you will be able to define homologous characteristics, define analogous characteristics, analyze the tracing of evolutionary relationships with examples, explain tracing the human evolution, define a fossil, and identify the methods used to find the age of fossils. Characteristics are the hereditary traits transmitted from parent organisms to their offspring. They can be related to particular appearance or behavior. For example, we have 4 limbs. That's a characteristic. Preparation of food through photosynthesis is a basic characteristic of plants. Characteristics in far related organisms would be similar because they are inherited from a common ancestor. Such characteristics are called homologous characteristics. For example, mammals, birds, reptiles and amphibians have 4 limbs with the same basic limb layout because they have inherited the limbs from a common ancestor. These limbs have been modified to perform different functions. However, all similarities between animals are not necessarily because of a common ancestry. Let's consider the example of bats and birds. These 2 animals have wings, but if you take an example of squirrels and lizards, they do not have wings. From this, we can conclude that birds and bats are more closely related to each other when compared to squirrels and lizards. The wings of bats have skin folds stretched between elongated fingers, whereas the wings of birds are feathery, covering the arm entirely. The designs of the 2 wings look similar because they have a common purpose: to fly. Their structure and components, however, are different. Therefore, although their origin is not common, they serve similar functions. This makes it an analogous characteristic rather than a homologous characteristic. Thus, homologous characteristics and analogous characteristics help us trace evolutionary relationships. They are evidence of evolution. Studies on characteristics were not only conducted on the current species, but also done on the species that are now extinct. How can we know that an extinct species ever existed? When an organism dies, it gets decomposed and leaves behind remnants in the soil. These traces of living organisms are known as fossils. Let's illustrate the process of fossilization of an ammonite. On its death, and ammonite sinks to the sea floor. Scavengers feed on the fleshy body, and all that remains is the shell. Gradually, the ammonite's shell is covered with silt and sand. After a few hundred years, the shell gets buried several feet beneath the surface of the earth. Gradually the shell decays, and what remains is a rock-like copy of the original shell. This is exposed when the rock is eroded by the wind, water, ice, or by the activities of paleontologists. Let's take a look at various fossils. Fossil trilobite, an invertebrate animal. Fossil fish. Fossil tree trunk. The dinosaur skull. This fossil was found only a few years ago in the Narmada Valley. Mary Anning found the specimen of Ichthyosaurus and was the first fossilist. Evolution doesn't happen in a single day. It is a process which takes place in stages. For example, complicated organs such as the eye are not generated by a single DNA change. The eye was formed over generations, starting from the intermediate rudimentary eye in planaria to the well-complexed eye in vertebrates. All of these changes are in response to adaptation to the environment. For example, the eyes of the octopus and the eyes of vertebrates have evolved entirely independently. These similarities of structure, despite different origins, provide a classic example of biological convergence. A property that is characteristic of a particular animal may, post-evolution, be useful for performing a totally different function. For example, long feathers were considered to provide insulation in cold weather. Some reptiles like dinosaurs had feathers, but very few were adapted for flying. In the present day, birds use feathers for flight, which is an example of adaptation. This means birds are closely related to reptiles. Thus, we can say that adaptation is an evidence of evolution. There are also many characteristics to prove evolutionary relationships in plants. In the present scenario, farmers use plants with desirable characteristics to produce new varieties by a process called artificial selection. Farmers have descended all these vegetables from the wild cabbage ancestor. Short distances between leaves gave rise to a new variety called cabbage. Arrested flower development gave rise to a new vegetable, broccoli. Sterile flowers gave rise to the cauliflower. Swollen parts gave rise to kohlrabi. And larger leaves gave rise to a leafy vegetable, kale. These changes are observed due to changes in genes which accumulate over a period of time. The tools for tracing evolutionary relationships, like excavating, time dating, studying fossils, and determining DNA sequences have been used for studying human evolution. All humans are a single species. The earliest members of the human species, Homo sapiens, are traced to Africa. A couple of 100,000 years ago, some of our ancestors left Africa and slowly spread across the planet. It resulted in stopping the gene flow, and over time it led to the evolution of different races. It is very crucial to understand certain things in tracing evolutionary relationships. Firstly, multiple branches are possible at each and every stage. For example, it is not true that human beings evolve from chimpanzees. Instead, both humans and chimpanzees had a common ancestor. Changes in DNA during reproduction are the basic events in evolution. Comparing the DNA of different species allows us to estimate how much the DNA has changed during the formation of these new species. In fact, there is no real progress in the idea of evolution. Evolution is simply the generation of diversity and shaping of the diversity by environmental selection. The only progressive trend in evolution seems to be that more and more complex body designs have emerged over time. However, many old and simple designs still survive. For example, one of the simpler forms, bacteria, is able to survive in almost all climatic conditions such as hot springs, deep-sea thermal events, and in Antarctica. In other words, human beings are not the height of evolution, but simply yet another species in the crowded spectrum of evolving life.